I think it's 9:30 and good morning everybody and welcome to the EASL Congress 2023 Vienna official press conference. My name is Aleksander Krag. I'm the Vice Secretary General of EASL and it's my pleasure to moderate this session. And also a warm welcome to you from media representative both online and also on-site. I think we have an outstanding number of new knowledge and science coming through, and hopefully this is also something you will help to bring to the broader public, which I'm sure they would be very interested in. You know, before we start, I just have a few remarks of how we see liver disease today. You know, it's largely seen as a silent disease. It often emerges late, maybe too late. Patients die, we cannot do much, but we are seeing really a changing landscape. Yet we can see it's some of the fastest growing diseases, for instance in UK, a leading cause of death among younger adults, 35 to 39. And also the fastest increasing reason for hepatocellular carcinoma, lead driver of liver transplantation, fatty liver disease in US. And more than that, the underlying cost of these diseases is massive. Alone in Europe, it's estimated to be 35 billion a year in direct cost and maybe up to 200 billion in indirect cost, because many of these diseases attack people at an early age, meaning that their working life year lost is massive if you die around 50. And yet now, if we just look at fatty liver diseases, you know, 30% of the adult population and 10% of the kids have it, which is really alarming. So obviously prevention is vital. We don't have much today about policy, but obviously things like policy change, let's say banning advertising of unhealthy food and alcohol to kids would be a good start. But prevention cannot solve all the problem. So I think we need to go forward. But before, I just want to let you know that before I introduce my colleagues here, that we are streaming live and we will also show it on EASL Campus and YouTube. So I hope you have a lot of good questions. I'll also maybe ask some questions to my colleagues to unfold a little bit so we're sure we have a very good perspective on what this actually means. So I guess we're interested in how that impacts our patients, how it impacts society, what we're doing here. So, but please, when you do it, then indicate your media outlet and then direct your questions. And I think the same for those online, and I think I have an iPad here that's supposed to send me the questions. And then just one brief thing. So we have an empty seat down there. That's Maria Buti, and that tells us how busy people like us are at the moment. She's chairing a big session in there, but she will join us and she will go last. So, but otherwise, I will present our first speaker, Professor Stephen Harrison. And Stephen will tell us something we I think been waiting for a long time, and he will later at our general session share the full data set. I think it's really, I think this is a potential game changer in the field of fatty liver disease. It's a phase 3 trial with almost 1,000 patients with 52 weeks treatment and dual biopsy. So, Stephen, please tell us what this means.
Sure. Thank you, Aleksander. It's good to be here to be with you guys today to tell you a little bit about the trial I'll be presenting later today. And the setting of this is, it's all about NAFLD and NASH. NAFLD and NASH is really an epidemic. It impacts about 25 to 30% of our population worldwide. And among that population, there's about 25% that have NASH, or the type of fatty liver that progresses and develops fibrosis and can lead to a negative outcome. With that in mind, resmetirom is an oral, once daily, liver-targeted THR-beta agonist. And in the setting of lipotoxicity, thyroid hormone function mediated through THR-beta is disrupted, impacting lipoprotein metabolism as well as beta oxidation of fat in the liver. And in patients with NASH, selecting for THR-beta may provide metabolic benefits of thyroid hormone that are mediated by the liver to include reduction of excess hepatic fat and atherogenic lipids. Maestro-NASH is a double-blind, randomized, placebo-controlled phase 3 trial evaluating the efficacy and safety of resmetirom in adults with biopsy-confirmed NASH. Today, we report week 52 data in 966 patients with NASH and F1B, F2, and F3 fibrosis. There's a dual primary endpoint established by both regulatory authorities for NASH resolution without worsening of fibrosis and fibrosis improvement of at least one stage without worsening of NAFLD activity score. I'm proud to tell you today that both endpoints were achieved. NASH resolution, 10% for placebo, 26% for the low dose 80 milligram, and 30% for the high dose 100 milligram. Fibrosis improvement was also met, 14% placebo, 24% 80 milligram dose, 26% 100 milligram dose. In addition, fewer patients progressed from F1B2 to F3. These data are supported by a late breaker poster we had here looking at AI digital pathology. And there, the data were very supportive. A one-stage improvement utilizing that tool was a 22 to 24% treatment effect delta, a two-stage improvement, a 12 to 18% treatment effect delta, and consistent with the data I just told you, a third less fibrosis progression. When we look at people that had liver biopsies at both time points, at baseline and at 52 weeks, this is more of the MITT population. Looking at NASH resolution, the treatment effect delta for the high dose was 28%. When we look at fibrosis improvement using that same analogy of a biopsy at the beginning and everybody having a biopsy at the end, the treatment effect delta was roughly 17%. In addition, there's a key secondary endpoint, which was LDL lowering, that also met statistical significance, 14% lowering in the 80 milligram dose and 16% lowering in the high dose. There were also significant effects on multiple atherogenic lipids in this population. Importantly, also consistent with that histopathology data, liver enzymes all significantly improved. Our non-invasive test, as measured by MRI proton density fat fraction, which is the gold standard for fat quantification in the liver, was significantly improved, as did liver and spleen volumes. There's a very positive effect also on FibroScan kilopascals. Importantly, safety, critical for this type of trial, was looked at very, very closely. And I can say the drug is very safe and well tolerated. The treatment emergent adverse events were balanced across the groups. Most were mild or moderate, and the most common side effect was GI related. So in conclusion, this is the first treatment to achieve meaningful effects on both primary liver endpoints, reasonably likely to produce clinical benefit in a phase 3 trial in patients with NASH. Both doses were effective. Multiple supporting analysis and additional data using non-invasive tests provide confidence in the biopsy data that I told you about today. The safety profile is excellent. Most common adverse events were GI-related and mild to moderate and tended to occur early in the course of therapy with a duration of approximately 2 weeks. The data support the potential for resmetirom to provide benefit for patients with NASH. Thank you.
Thank you very much, Stephen. I think this is really, really exciting with this field, really, let's say, breakthrough, game changer. I guess that's kind of the words, but we can come back to that in the discussion. And now to the next speaker is Lindsey Edwards from King's in London. A completely different approach to a different stage of disease, yet where there is, again, a huge unmet need. But how does the bugs within us interfere with how we live and thrive, and how can you modulate that? So over to you, Lindsey.
Thank you very much. It's a pleasure to be here. So I'm going to take you right back to the beginning of time. So I don't know if you know, but there are more microbes on Earth than there are stars in the universe, or grains of sand on every single beach on this planet. So all of those microbes interact with us on our mucosal surfaces. The greatest concentration of those is in the gut. So to stop those microbes getting into our body and causing us damage, we have a single cell epithelial layer. And we have a huge immune system in our gut to try and stop those bugs from translocating across. So if they do get across, they drain to the liver, and the liver is your last resort to deal with that before it gets into your body and causes a systemic infection, which quite often is lethal. So we have this interaction with our immune system and these bugs, and quite often that goes wrong in liver disease. And then with the liver disease, you don't have the right immune system to fight it off, so your immune system is defective. The problem with that is that then you end up getting all of these pathogenic bad bacteria that go into your body, into your liver, and cause infections. With that, those pathogens make an environment in the gut that doesn't support your good and friendly bacteria. You need those bacteria because they have a whole set of enzymes that digest all of the food that we eat. If you don't have those bugs, you can't digest the food properly, you don't have the right energy. That also means you don't have the right energy for the cells in your gut to defend that barrier or for your immune system. So we need those bugs and we need those enzymes. Essentially what we have done is something called fecal microbiota transplantation. It's pretty crude. We take poop or poo, we mash it up in a cocktail blender. I'm not sure if really this is, we joke and say shaken, not stirred, but you don't drink it. It's either administered by endoscopy or you can take it by capsules, or we call them crapsules. So you take these and basically it restores all of your good bacteria. But the good thing in restoring those good bacteria is it restores all of those enzymes and it metabolically reprograms you, essentially. And in that metabolic reprogramming, what happens is you give your immune system and your gut barrier all the energy that it needs to defend that barrier and stop those bugs from getting in. What that entails then is you actually reduce the incidence of infection, and infections to liver patients are deadly for two reasons. They either die of the infection or they get delisted from transplant, which is their last resort. So either way, if you get a deadly infection as a liver patient, you're most likely going to die. So it's really important to try and stop these patients from getting infections. What we're doing is basically bolstering your immune system so you don't need as many antibiotics. That's also very important because one of the largest global health problems alongside liver disease is antimicrobial resistance. So by boosting your own immune system, you don't need to take as many antibiotics, it benefits you from helping your liver disease, but it also benefits to stop the use of antibiotics. I probably stop.
No, that's absolutely beautiful. I'm sure you could continue, and we would love to hear about it.
Yeah.
Again, I think a beautiful example of how deeper understanding, change in technology can certainly lead to that we have whole new avenues of how we can understand and help patients in new ways. So I think we'll definitely come back to that. That's really brilliant and very complimentary from what we heard. The next thing is then another thing where the world is really moving and moving very fast now is AI, ChatGPT, and there are many, many other things. But what does that mean in hepatology? What are the pros and the cons? And if you not noticed, there are several abstracts out there, 25 this year. Let's see how many there will be in 5 years, probably 10 times as many, I imagine. So the question is, what can we use it to? How can that predict outcomes? How can we predict relations to drug effects? And there I think we're so privileged to have Julien Calderaro from France here who looked into that, into how these can predict outcomes around different drug classes. So please.
Thank you. So my medical specialty is pathology, and basically what we do is that we look at the tissues that, for example, we obtain through biopsy or surgical resection, and we look at the tissue, we review the overall architecture, and the tissue section is on a glass slide, and we use to review these slides using conventional microscope. And the specialty of pathology is now undergoing a major revolution because we have the ability to transform these glass slides into digital files. And you can view these digital files on a computer. And this is what explains that we can apply artificial intelligence to these digital files that are basically like images and pixels with values. And we can process all these values, which correspond to the tissue, the overall architecture, the appearance of the cell, by artificial intelligence in order to extract information that may not be accessible to the human eye. And we apply these kind of approaches to predict response to the atezolizumab-bevacizumab combination, which is the standard of care for patients with hepatocellular carcinoma, which is the most frequent primary liver cancer, which are the patient with this cancer with an advanced disease, which is not amenable to curative therapies. And the problem is that so far you give this treatment to every patient. Some of them will benefit, and some of the patients will not benefit from the treatment. They may have side effects, and maybe for this patient we may try other kind of drugs. And the overall problem is to predict which patients will benefit from the combination. So there has been some publications about molecular signatures that may help to predict response, but to do that you need to have an expertise in molecular biology and also bioinformatics. So you need to process the sample, you need to do complicated analysis, and this is very, very difficult to implement in clinical practice. And so far, for example, there is only for breast cancer for which we have a validated gene signature, which is a molecular biomarker. So what we try to do in the study that will be presented on Saturday is to predict using these digital slides, which is relatively easily available in hospitals, to predict this kind of molecular biomarker using artificial intelligence applied to histological slides. And we developed this model that is able to tell you just from the image of the tissue if this tumor overexpressed this marker or not. And we validated that in a large cohort of samples and a large cohort of patients that were treated by the combination. And importantly, also, we show that when we apply this model, this artificial intelligence model, in samples of patients who had a biopsy and that were further treated by the combination, we are able to predict which patient will benefit from this particular combination. And so if it is completely validated and provided this kind of validation, we may think that we could use this type of easy and really low-cost biomarker, because this prediction only takes a few minutes by artificial intelligence, to really give the right treatment to each patient, and maybe when we predict that the patient will not respond, immediately switch to another treatment regimen.
Thank you very much, Julien. I think, again, a brilliant example of how technology and medicine, they merge and can really make a big leap forward. Imagine today we know that not all patients for sure respond to a treatment. If you already up front know who it works with and who it doesn't work with, I think this really holds huge potential. And I guess also in a very pressed healthcare system all over the world, I think these are some of the technologies that can really help us solve it and still deliver the best patient care. So in the end, it's all about the patients. These are the ones we want to help. So therefore, we need the patients' voices. So now we heard some examples of advances that will improve and impact the way we see, the way we handle and manage diseases. But therefore, the patient's voice is really important. So Milan Mikoviky from the European Patients Organization, ELPA, is here. So Milan, please give your perspective.
Thank you very much, Professor Aleksander. Thank you very much to everybody present today. First of all, in the beginning, I will say a couple of words that it's my big honor to be here among such great scientists, which are somehow providing the wider perspectives for us like a patients, giving a hope, trying to resolve our, let's say, the biggest unmet needs which exist in the moment, and what is the most biggest challenges which are coming for the future for the liver patients. Like Aleksander mentioned, I'm coming from European Liver Patient Association. That's been an umbrella organization. We have 32 members from 25 European countries. ELPA is involved in a lot of different kind of processes. First of all, I'd like to thank you to EASL and Professor Krag that ELPA is somehow a unique organization. From his existence, is partnering with EASL the past 18 years, which give us opportunity to say that somehow on the table we feel that we are equal partners in some of the biggest research what are coming or what was done in the past. From a patient perspective, I will say that our expectation from all these, let's say, abstracts and research which are on the table in the moment, give me a pleasure that I hope so that we, like a patient, we will have the benefits and beneficiaries for the next period that our quality of life will be improved a lot. How mentioned Professor Aleksander that everything in the liver field is ongoing very fast in the moment. Like a patient advocate, I will say that we are struggling with different kind of approaches to supply access to our patients, to change the policy work around Europe and everything, what is connected for better and quality life for our group of patients. What is most important that ELPA is struggling in the same time in different kind of institution on a European level. We are part of European Commission's, let's say, bodies like DG Sante, ECDC, European Medical Agency, HERA, and all of these committees which are existing in the moment, which are giving for the first time somehow bigger visibility to stress the problems which liver community and liver patient are struggling in this moment. I will say a couple of words about the research which are on the table in the moment, which giving me a huge pleasure that in the same time, together with EASL and with the scientists of this eminent, let's say, hepatology group, give us possibility for ELPA to participate in the moment in more of 16, 17 research project, which this is quite beneficiary for us, how to approach and how to, let's say, develop a different kind of mechanism to this wider of clinical approaches to the patient group on national levels, and to help them how to develop all national strategies, all to be disseminated on right way. And what we are expecting, I will say, from all of this research, of course, is that the improving the quality of life and giving the fastest response. Early detection is very important. Stuart and Lindsey mentioned about the new achievements which are coming very fast about one of the, let's say, prevalent disease for NAFLD-NASH, which is for us something devastating like a patient organization, because still it's very difficult to be detected on the field. And we're struggling with the fast and early detection, and this giving a lot of possibilities now with this new research and approaches, this kind of patients to be, let's say, discovered. We like a patient, we're struggling for the right everybody to be detected and to know the diagnosis.
So sorry to interrupt you, Milan. No, it's lovely,
Sorry.
But I think we will also just make sure we have enough time for questions, and I think we will all be available afterwards, so then we can drill into it and also have this discussion. So sorry for interrupting you, but now Maria is done with her session, so welcome to you also, Maria Buti, who is chair of the department in Vall in Barcelona, but also serves as the chair of the Public Health Policy and Advocacy Committee at EASL. She is a world-renowned expert in hepatitis B. And I think, Maria, you have got something new on how we should actually manage the use of tenofovir and other drugs in hepatitis B, so please.
Yeah, thank you, Aleksander, and sorry for being late. As you know, hepatitis B infection is the most common viral infection that produces liver disease worldwide. There are almost 300 million of individuals living with chronic hepatitis B. And those individuals are at risk of developing cirrhosis, decompensation, and liver cancer. We have several drugs that are very useful to prevent decompensation, development of liver cirrhosis, but we don't completely abolish the risk of liver cancer. So with the current drugs, we have tenofovir and tekavir, and the last drug is TAF. The experience with TAF was very limited. And the abstract that I am going to present tomorrow discuss the longest experience with this new drug, that it's TAF. It's similar, it's a prodrug of tenofovir with the advantage to have a better safety profile. And in this study, we explore in a large number of patients, more than 1,000 patients with chronic hepatitis B in different centers globally. The use of TAF during the first 2 years, it's a double-blind study that compared TAF, this new prodrug of tenofovir, versus the old formulation, tenofovir, for 2 years. And now, tomorrow, I am going to present the open-label phase that is extending the treatment duration until 8 years. The patients included in this study are those typical with chronic hepatitis B, that mainly men, median age, different HBV genotypes, high viral load, elevated ALT. That means patients with liver damage. They received the drug for 8 years. And we look at different endpoints. The first endpoint, that it's a very important endpoint, is virological suppression. And during all of these 8 years, the majority of patients achieve virological suppression. That it's good, because virological suppression is associated with less risk of developing cirrhosis and decompensation. The second endpoint is an ALT normalization, liver enzymes normalization, and it's achieved in the majority of patients. However, despite this virological suppression and normalization of the liver enzymes, the majority of patients persist with HBs antigen positive. That means that there is still infection, still hepatitis B. This is very similar with the all drugs, and clearly shows that hepatitis B is very difficult to eliminate, to control, and we need new drugs for these patients. And regarding safety, will be a poster that looks at the safety of TAF and the safety of TAF during this double-blind phase is better than tenofovir, and during the open-label phase, the safety is maintained. So I think in summary, these are the main findings of this study of the use of TAF in patients with chronic hepatitis B. And this study included 2 types of patients, E positive and E negative patients. And the majority of our patients here in Europe are E negative patients. So I am happy to...
Yeah, no, thank you very much, Maria. I think it's beautiful. Again, another disease class, hepatitis B. It's a huge population all over the world. Then again, explaining, I think, a real step forward, better safety, and still acknowledging we are not there yet, but something that will definitely impact patients in a good way. So I think now we're done with the formal presentation, and now the floor and online open for questions. So please come with your questions. I can maybe start then with one just to get it going. Oh, yeah, no, please, yeah, Medscape, yeah.
I'm Ed Sussman with MedPage Today. Dr. Calderaro, has anyone actually used this AI algorithm to manage patients with disease? Or is this just something that's, and if not, what are the next steps in order to make that a possibility?
So it's a very, very good question. So our study was like a proof of concept study. We wanted to show that it was possible to do this kind of models. There are several issues that we have to overcome to make this completely available in daily practice. First, we have to have pathology departments that are completely digital, and it is not the case yet. For example, in France, there was about 12% of the pathology departments that are digital. And I think we still need to improve the models, and I would like to underscore that the main issue now is the access to data, because the technology, the AI technology is there. You can do very fancy models with a computer that costs a reasonable amount of money. But the main problem is access to data, and this is our main issue, and we are struggling to get access to data from hospitals to make really highly efficient model. And I think this is the main issue before thinking of implementing this kind of model into practice. But definitely the technology is there. I think it's everywhere in Facebook, Twitter, when they tag your face, and so it works. We know that it works. And now we have to meet all the regulation standard. We have to assess the models with really rigorous methods. But I think it will take still a few years before we are completely ready.
Thank you. And questions for Dr. Edwards. What were the outcomes with the 32 patients in the pilot study?
So in terms of clinical outputs, we're only following the patients for 90 days, but we actually do see an increase in albumin, and we see a reduction in hepatic encephalopathy score, which is fairly impressive, really, if you think we're only following them for 90 days. So what I can tell you is that we restore the gut barrier in about 7 to 30 days. So that's not likely in 90 days to translate to huge clinical outputs anyway. But we are we've just started a PROMISE trial, which is a larger version of this. So it's 300 patients, 17 centres. We will follow them up for 2 years. They will get dosed with the capsules or crapsules every 3 months for the course of the 2 years. So there we will be able to tell whether or not we really do see a huge impact in clinical outcomes.
Did you see a successful transplant in all 32 of the patients?
Yes, but there is variation. So what we're looking for is engraftment, something called engraftment. So there is a whole host of things that determine what with engraftment. Your host immunity, your donor immunity, your donor diet, your host diet are all factors that play a part in this. Antibiotics is a big thing. So there's a huge bone of contention of whether do you give antibiotics before you do the FMT, do you not? In our case, we didn't give antibiotics, including rifaximin, so they weren't allowed to enter the study if they did. But unfortunately, there were a couple of patients that became unwell, not because of the FMT, something completely independent of that, who had to have antibiotics, and of course that wipes out whatever we had put in. So there are some patients that had better engraftment.
Sorry again to interrupt, Lindsey. I think the more detailed, we keep the answers and the question relatively short, and then we can expand afterwards, because I think we have more questions. So if that's okay, then I think, please
go ahead.
Unless you have.
You have more? Or,
I've got one more quick one for.
These are interesting for me too.
Yeah.
For Dr. Edwards again. How do you determine whose poo you should use?
Oh, now that's a really great question. So we have, super donors of what people are looking for. So from the work that we've done, actually what we're looking for is something that induces something called an omnivorous diet. So you do need some complex carbohydrates, and you do need some protein. So you need the bugs that can metabolize both of those. So our patients that received the FMT were mainly had a Western diet, so high sugar, high fat, high animal protein. The donors happen to be all vegan or vegetarian, but it's not that we're suggesting that you should be vegan or vegetarian. It's because you had then a mix of the ability to digest your protein and the ability to digest your carbohydrate. So that's what you really need is an omnivorous diet.
Thank you, Lindsey.
Thank you. Actually, to build up on that question, I'm really glad you had that last one. I was wondering about safety, and will the stool also be filtered for microbes just to avoid any unwanted bugs that don't harm a healthy donor but possibly a diseased patient?
Of course, yes. So they're heavily screened for, well, it was 26, I think it's now even more. We've obviously had to add in COVID as well. So they're heavily screened to check. They also undergo, lots of different testing, lifestyle questionnaires, all of that kind of thing, to make sure there is nothing that we're transmitting across.
No option to filter, like just select some microbes that you know are beneficial, which would be much more advanced, I guess.
You would say that, but what we actually think is happening is that you're transplanting a stable community. So obviously the ideal world will be that we would be able to select a few bugs and that's what you transplant. I think it's looking to be more complicated than that, that it's all of the species together in the right community, the right metabolites, all of these things, and that's what we're transplanting. But we're working on trying to find what's in it.
Thank you. Another question to Stephen Harrison. Very interesting data. I'm getting more hopeful that we will finally see some advances in NASH. If everything goes perfectly well, that's the first question, when would you expect this to become available to patients? And then on a broader picture, because I know you've been in NASH for many years and you're overviewing the whole field, there's lots of other liver diseases with overlaps. Viral hepatitis does not prevent you from having NASH, PBC doesn't. Do you see any issues combining future NASH drugs with medicines for these other diseases? Are there any signals? Are there any plans for studies?
Sure. So just to your first question, so, I think that this is an exciting time. It's an exciting time for the field of NASH in particular. And to be on the forefront of having a potential drug to treat our patients with disease is something that I've worked for the better part of 20 years. So it's outstanding to be here. So where do we go from here? I think the company has publicly announced a plan to submit a new drug application to the FDA by the end of the second quarter. They do have breakthrough status designation with the FDA and with a subsequent priority review, potentially that could be reviewed in 6 months. So we're looking, hopefully towards the first part of 2024 to have that drug available, best case scenario. To your second point, I think absolutely these drugs, these metabolic drugs potentially have benefit on other dysregulated energy metabolism problems, such as cardiovascular disease. If you look at resmetirom, it has very good results on atherogenic lipids, and we don't see any drug-drug interactions. With other liver diseases, it hasn't been studied directly. We've intentionally excluded other liver diseases in the setting of these trials, but I don't see any issues, particularly like with hepatitis B therapy or chronic immunosuppression for autoimmune hepatitis. I think that would be fine.
Thank you.
Can I add a comment regarding hepatitis B? In this large study on hepatitis B using TAF, there is a small subset of patients who did not normalize ALT levels. And these patients are completely viral suppressed, and the reason to maintain elevated ALT levels are diabetes, obesity. So clearly, we need also for this subset of patients with chronic hepatitis B that have metabolic liver disease, these new drugs.
I mean, this is something we see in cured hepatitis C patients also, who don't normalize the enzymes. I actually have a question for you too about safety. Is it better than TDF all around, or is there any issue that is there with TAF and not with TDF? I once heard something anecdotal about more headaches, but I'm not sure if that was ever substantiated.
Yeah, thank you for the question. Overall, the safety profile is better, but it's particularly better for a subset of patients that the EASL guidelines, the 2017 EASL guidelines, have been identified, that are patients that have bone alterations like osteoporosis, and also for patients with renal disease. And one of the advantages of TAF is that it's given at a fixed dose, doesn't need to be adjusted to renal function like entecavir or the prodrug tenofovir. And I think the best advantage is for this subset of patients that EASL guidelines identify. But looking at hepatitis B, that is predominantly a disease of low and middle-income countries, having a drug that needs less monitoring, I think it's an advantage.
Thank you. If there's nobody else, you have a question.
Yeah. I also have a few online questions, so maybe we're coming a little to an end,
please.
so brief questions, brief answers. So I have two here for you, Stephen, here from Hugh Thomas. Very exciting data. There's two questions. Did you see any difference in tolerability between the two doses, and were discontinuation balanced versus placebo?
Yeah, so the tolerability was essentially equivalent between the two doses. As far as treatment emergent adverse events that led to discontinuation, there were a few more in the 100 milligram dose that were GI related. The GI related adverse events tended to be diarrhea and nausea. If you look at the electronic data capture system, about half of the diarrhea cases were worsening of previous diarrhea, intermittent loose stools, tended to occur immediately after dosing. Duration was about 2 weeks and no difference between the two doses. Nausea tended to occur more commonly in women. Again, at the start of therapy, tended to wane after about 2 weeks or so. When we look at beyond 52 weeks of therapy, there were no discontinuations really to any significant degree in the high dose group.
Okay, and then another one, were there any effects on resmetirom on body composition, and was that assessed?
Body weight was assessed, not body composition, so there's no impact on body weight, glycemic control, or glucose.
Okay, and then the second one here is from Madeleine Armstrong, again for you, Stephen. Can you explain the rationale behind changing the primary endpoint from looking at NASH activity score to NAFLD activity score?
I'm not sure I understand that question. So the primary endpoint was a dual endpoint, NASH resolution with a 2-point improvement in the NAFLD activity score, same that we had in the phase 2, without worsening of fibrosis. I don't think that's
changing during the course of the trial. I don't know.
No,
Okay.
we didn't change that during the course of the trial. She may be asking about fibrosis improvement without worsening of NAS versus without worsening of NASH.
Yeah,
so
that's probably,
So in that situation, again, we stayed consistent with the phase 2 data. And when you think about it, in the setting of the phase 2 data, where you had fibrosis improvement, we didn't have worsening of inflammation or ballooning. That just makes sense. As disease severity, you wouldn't expect fibrosis improvement if disease severity is worsening. So again, interpreting the guidance from the FDA in 2018, and no pushback from meeting with the FDA relative to that endpoint, that's what we went with.
Thank you very much. Yes, please.
Hi, it's Becky from Medscape. Question for Stephen. I'm really just want to get a feel for what this means for the clinician and for the patient. And perhaps, Milan, you could come in there on that side, the latter. Where does this, what sort of unmet medical need does this meet? It's to get a real feel for, I can get the data, the numbers at your presentation, that's fine. It's just getting a really nice comment on what this difference this will make to practice and patients.
Huge difference. I mean, heretofore, no approved therapy, lifestyle modification, and we know patients are challenged with the ability to lose weight and keep the weight off. And the impact on changing their lifestyle as well is something that's very, very hard to do. What I tell patients is, unless you have heart transformation, you can't have behavioral modification. So therapy has been needed for a long, long time. There's been a delay in identifying patients with this disease because many of my GI colleagues say, until you have a treatment, we're not going to work these patients up for this. So now that we are on the forefront of a treatment, it allows clinicians to begin to open their minds about the possibility of identifying these patients. So I think, having a therapy that could mitigate both the disease activity and the fibrosis is absolutely critical. Fibrosis portends a worse prognosis. So where we can mitigate that, I think we'll have what I like to say is we leave a big fat dent on the planet in the name of NASH.
Thank you.
And your trial had an 80 milligram and 100 milligram, so do you go for both of those doses?
Yes.
You do?
Ideally, I mean, both met the endpoint, so you would have, 2 doses to consider.
Okay, fine. And
Milan,
Milan,
the patient
patients,
perspective.
Thank you very much for the question. Really, it's huge, I will say the same, because the hope is one of the most things what is important for us like a patient. We see a hope in this research that suddenly we will get a drug who will somehow, let's say, improve all quality of life, which till now a lot of patients can manage the lifestyle. We know with the new era of technology and everything what is going on in the process of the life in the moment everywhere is that the fast life not giving a lot of possibilities to people to approach let's say good style of life. That means that not everybody can approach to healthy food, first of all. The first step depends on the countries, developed, non-developed, middle-income countries or something like that. The third part, if we have a possibility to have a drug who will, of course, be affordable, accessible for us, and not expensive so much, then we are reducing a lot, especially in the field of the, let's say, the future liver cancer projection, which was very high till 2040. That means for us a huge opportunity how to, let's say, manage the next expectation for the patient and patient groups, case managers and caregivers for the future. I think this is a huge improvement in let's say, this field for liver diseases, which giving me pleasure to say that very fast it's going to be the approach through different kind of, let's say, directly, indirectly diseases or conditions which exist in the moment, like obesity or something like that, to approach these medicaments and access of affordability.
Thank you very much, Milan. I think we're running out of time, and I know at least Stephen needs to go because we have the general session and you will present it in 13 minutes, so you better be fresh. Any final questions? Otherwise, I think the rest of us will stay around if you need something. But I think, Stephen, if you are free to go, we prefer you're fresh down there. And I guess you can reach out to him later if you have additional questions. But please.
I was wondering what the price point of TAF or TAF would be. I know that for TDF they've recently reduced the price quite significantly, so I'm wondering if it's going to be sort of available on that sort of scale and what the price point of the treatment would be.
Sorry, I missed the first part of your comment.
I was just wondering what the price point of TAF would be. I know that they've recently reduced the price of TDF quite significantly to make it available to more patients, so I'm wondering what it would cost and whether it would be available widely to lots of patients or if it's still...
Yeah, up to now in some countries the cost of TAF is higher than TDF in some countries. In others have a similar price. I think the big difference is the TDF is a generic drug and it's available in the majority of countries, and TAF probably will be available in the next years as a generic.
Okay, thank you very much. Thank you very much for joining. Thank you very much for your questions. And again, we'll be available to support you further. But thank you very much, and we will close it here.